Good morning students, and we are continuing with unit number 1, that is metabolic pathways in higher plants and their determination. And I already told that this unit is divided into two different portions. In the first part, you have to see about various metabolic pathways like shikimic acid pathway, then acetate mevalonate pathway, then acetate malonate pathway, amino acid pathway, etc. That we already completed. And in the second part, you have to study about the investigation of biogenetic studies. That is how these biogenetic pathways are means taking place inside the plant. That is what we are going to study in the second part of this unit. Okay, so in your syllabus it is mentioned like this: study of utilization of radioactive isotopes in the investigation of biogenetic studies. So that is what we are going to study under this unit. Okay, so we will be finishing this unit today in two videos. In the first video, you will be seeing something about the tracer techniques, and in the second video, you will be seeing about the remaining portions of tracer techniques. Okay, so that's all. അപ്പോൾ ഇത്രയാണ് നമുക്ക് ഇതിനകത്ത് പഠിക്കാനുള്ള സംഭവങ്ങൾ എന്ന് പറയുന്നത്. Okay, so now we can enter into the chapter. So the topic is like this: study of utilization of radioactive isotopes. That is, study of utilization of radioactive isotopes in the investigation of biogenetic studies. Okay, and to your university exam questions, means in your university exam, the question may be asked like this: explain about tracer techniques. Then what are the various methods involved in the investigation of biogenetic pathways? Then third thing is utilization of radioactive isotopes in the investigation of biogenetic studies. So these are the possible questions that you can expect from this topic, and the answer of all these three questions are the same only. So if the question is asked regarding tracer technique, then you have to write only about the tracer techniques. That's enough. Okay, it may be either asked for a 5-mark question, or even it may be asked for 10-mark essay question. Clear? And sometimes if the question is asked like this: what are the different methods involved in the investigation of biogenetic studies? Then you have to write the entire thing. That is, you have to write about tracer techniques and also about the various other minor techniques that involved in this studies. Clear? So that is what you are going to study under this topic. Clear? So before that, first we have to see what are the things we are covering under this unit. So first thing is about biogenetic studies. So we are hearing about the term biogenetic studies. So first you have to understand what do you mean by biogenetic studies. Clear? So എന്താണ് biogenetic studies? അത് നമ്മൾ ആദ്യം മനസ്സിലാക്കണം. So first you have to understand that we already studied about different pathways. For example, shikimic acid pathway we already studied. Clear? And this pathway is a metabolic pathway, and here a starting compound is there. Likewise, some intermediate compounds were also there. Clear? And from these intermediates, some final products were also obtained, and in this pathway we got 3 different amino acids. In shikimic acid pathway, 3 different amino acids are biosynthesized, and they are tryptophan, tyrosine, phenylalanine, etc. So these are the 3 different amino acids that are biosynthesized in this pathway. Clear? So here in this section we are going to study how we understand these are the intermediates in that pathway. Studying about that is known as biogenetic studies. So അതായത് വേറെ ഒന്നുമില്ല, ഒരു ബയോജനറ്റിക് പാത്വേ എങ്ങനെയാണ് നടക്കുന്നത് എന്നതിനെ കുറിച്ച് പഠിക്കുന്നതിനെയാണ് ബയോജനറ്റിക് സ്റ്റഡീസ് എന്ന് പറയുന്നത്. Hope you understand the topic. Clear? Now that is the first thing you have to understand. Then the second thing is about isotopes. Means you have to understand what are isotopes. You already studied in your lower classes. What are isotopes? They are the substances which are having the same atomic number, but it differs in their atomic weight. Clear? So that is known as isotopes. And regarding isotopes, 2 different types of isotopes are there: stable isotopes and unstable isotopes or radioactive isotopes. So here radioactive isotopes means endana, they are having the capacity to emit radiation, that is alpha rays, gamma ray, beta ray, etc. Likewise, some radiation can be emitted by these isotopes, and they are known as radioactive isotopes. And in our section we are going to use this unstable isotope, that is radioactive isotopes. That is, radioactive isotopes were generally used in the biogenetic study of various plant compounds. Clear? So that is isotopes. Then the third thing is about tracer techniques. So ഇതാണ് exam ആയിട്ട് ചോദിക്കാൻ ചാൻസ് ഉള്ളത്. So what are the things you are going to study under this tracer techniques? So these are the things that you are going to study under this topic. So first you have to get an idea regarding biogenetic studies. What do you mean by biogenetic studies? So but you have to understand what is biogenetic studies. Means through which pathway? Means by a pathway only some compounds were synthesized inside the plant. And what are the intermediates, what are the starting material, and what are the final products that we already knowing, and how these compounds are synthesized, that we are investigating, and this investigation is known as the biogenetic studies. And here normally radioactive isotopes were used in the investigation of this metabolic pathways. Okay, so radioactive isotopes means they are having the capacity of emitting radiation. Before that, isotopes are those compounds which are having same atomic number but differs in their atomic weight. Clear? Okay, so and the final thing is about tracer techniques. So first we can see about, some other methods were also there. Apart from this tracer technique is very important, and apart from tracer technique, some other methods were also there in studying about the investigation of biogenetic pathways. Clear? So first we can see about this introduction. Means എന്താണെന്നുള്ള കാര്യം just ഒരു introduction ആയിട്ട് നമ്മൾ പഠിക്കുകയാണ്. So here you already knowing that plants are biochemical laboratories. Biochemical laboratories, laboratory you already knowing. In a laboratory lot of compounds were synthesized as well as lot of compounds were produced, etc. So likewise we can consider, say that plants were also biochemical laboratories, and in plants various secondary as well as primary metabolites are produced. So this you already studied in your fourth semester. What do you mean by primary metabolites and what are secondary metabolites? So here the secondary metabolites are the useful medicinal compounds. Likewise, some primary metabolites, for example, carbohydrates, etc., were also produced inside the plant. Okay, so plants are biochemical laboratories, and various secondary as well as primary metabolites were produced inside the plant. Clear? Now, next thing is, for the production of this primary as well as secondary metabolites, lot of intermediates are required. You already knowing that not means a compound is not directly formed. Okay, so for the formation of a compound, it requires the starting material. Likewise, some intermediates, lot of intermediates were also required in the production of a final compound. So here for the formation of this primary as well as secondary metabolites, lot of intermediates were required. Clear? And this study, that is the study of the pathways, study of this pathways is very complicated. So it is very difficult for us to understand what are the different intermediates which are taking place inside the biosynthesis of a particular pathway. For example, shikimic acid pathway. So starting material is phosphoenol pyruvate, then erythrosaur phosphate. Likewise, then after something one keto compound is there, then dihydroshikimic acid, quinic acid. Likewise, lot of intermediate compounds are there, and it is very difficult to find out what are the intermediates that are taking place inside in the synthesis of these compounds. Okay, that is the study of this pathway is very difficult. But nowadays, lot of techniques were available for studying about this metabolic pathways. And among the different techniques that are available, this tracer techniques plays a major role in the investigation of biogenetic pathways. Clear? That is, plants and biochemical laboratories, then various secondary as well as primary metabolites were produced, then lot of intermediates were also required in the production of this metabolic pathways, in the formation of this metabolic pathways. And the study of this metabolic pathway is very difficult, and nowadays lot of techniques were available, and among the different techniques available, this one, that is tracer techniques, plays a major role in the investigation of biogenetic pathways. Clear? So now other apart from tracer techniques, some other techniques were also there which which is helpful in the investigation of biogenetic pathways, and they are use of isolated organs and tissues. Use of isolated organs and tissues. Then the next thing is about grafting technique, and last thing is use of mutant strains. So these are the different techniques that are used in the investigation of biogenetic pathways. Okay. So we already knowing that plants are biochemical laboratories, and lot of primary as well as secondary metabolites were produced inside the plant. And for the production of this primary as well as secondary metabolites, lot of intermediates were required. And the study of this pathway that leads to the formation of this metabolite is very difficult. But nowadays different techniques were available for investigation of this biogenetic pathways. And the common techniques that are used in the study of these pathways are tracer techniques, then use of isolated organs and tissues, then grafting techniques, then use of mutant strains, etc. Clear? So among all these techniques, this one is very important, that is tracer techniques. And I already told that in your university exam, the question may be asked, write about tracer techniques for 10 mark or 5 marks. Okay, in sometimes the question may be asked, what are the different techniques that are available in the elucidation of biogenetic pathway? You have to write all the techniques, that is from tracer technique to use of mutant strains. In some other cases, the question may be like this. Explain about the use of radioactive isotopes in the investigation of biogenetic studies. Then you have to write about this tracer techniques. Clear? Okay. So in this video, you are going to see about tracer technique, and from the tracer technique, we are completing only two steps or two different techniques in this video. So first we can see about the tracer techniques. So like every other things, here also you have to write an introduction. So this tracer technique is the most widely accepted technology in the investigation of biogenesis or in the investigation of phytoconstituents. This is the most widely used technology, and here various labeled compounds are generally used. That is, in tracer techniques, various or different labeled compounds are generally used. And here normally unstable isotopes are used for this purpose. Labeled എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ means എന്തെങ്കിലും ഒരു മാർക്കിംഗ് നമ്മൾ കൊടുക്കും, അത് അറിയാൻ വേണ്ടിയുള്ള അങ്ങനെയുള്ള compounds ആണ് ഇതിനകത്ത് യൂസ് ചെയ്യുന്നത്. And here normally unstable isotopes are used for this purpose. Why we are using this unstable isotopes? Because here they were having the capacity to retain their radioactivity even after it is incorporated in the chemical reactions. So these isotopes or the labeled compounds which we are introducing will be interfering in the chemical activity or in the chemical reaction or in the metabolic pathway. But the radioactivity of the marked or the labeled compound which we are given to this pathway is not getting changed. So we can easily identify the compound which we already fed or the labeled compound. അതുകൊണ്ടാണ് we are using unstable isotope for this purpose. Clear? And now we can see about the various instruments used for the detecting of radioactivity, and they are, first one is ionization chamber, second one is Geiger-Müller counter, then the third one is scintillation counter, and the last one is gold leaf electroscope. So these are the different instruments used for the detection of radioactivity. What are they? They are ionization chamber, then Geiger-Müller counter, then scintillation counter, gold leaf electroscope, etc. Okay, so these are the common instruments which we are using in tracer techniques. Okay, then last thing you have to know about the different unstable isotopes that are used in the elucidation of this biosynthetic pathways. So we already studied that in this method, unstable radioactive isotopes were used for the studying of biosynthetic pathways. And the common unstable isotopes used in this biosynthetic pathway, means elucidation of biosynthetic pathway, are carbon 12, then carbon 13, carbon 14 is also there, then hydrogen 3 or tritium, then sulfur 35, phosphorus 32, then iodine 131, etc. So apart from this also, many other unstable intermediates or unstable isotopes were also there, but the commonly used unstable isotopes are carbon 12, carbon 13, hydrogen 3, carbon 14, sulfur 35, phosphorus 32, iodine 131, etc. So ഇത്രയാണ് tracer techniques in the introduction പറയാനുള്ളത്. എന്തൊക്കെയാണ്? It is the most widely used technology, and in tracer technique lot of or various labeled compounds are generally used. ഇനി അടുത്ത് എന്താണ് ഈ labeled compounds? Normally unstable isotopes were used as the labeled compounds. That is, a lot of labeled compounds were used, and unstable isotopes are generally used as the labeled compounds. Why we are using this unstable isotopes? Because even after getting incorporated into the chemical reactions, the radioactivity of these isotopes won't get altered, and it can be measured after the reaction. So the study of this means biosynthetic pathway makes easy with the help of unstable isotopes. That is the purpose why we are using this unstable isotopes. Okay. Now, the various instruments used for the detection of radioactivity are ionization chamber, Geiger-Müller counter, scintillation counter, gold leaf electroscope, etc. Then next thing is about the commonly used unstable isotopes are carbon 12, carbon 13, hydrogen 3, sulfur 35, phosphorus 32, iodine 135, etc. Clear? Now, means in tracer techniques, different types are there. So what it means, first of all, we already seen about an introduction regarding tracer techniques, but in tracer technique, different types of techniques were available, and among the first one is autoradiography. ഇതെന്താണ് നമുക്ക് പിന്നീട് പഠിക്കാം. Autoradiography. Second thing is precursor product sequence. ഇത് ഓരോന്നും ഡീറ്റെയിൽ ആയിട്ട് പഠിക്കണം. So first one is autoradiography. Second is precursor product sequence. Then third one is competitive feeding. Then the fourth one is sequential analysis. So these are the four different types of tracer techniques. Four different types of tracer techniques are there. They are autoradiography, then precursor product sequence, competitive feeding, then sequential analysis, etc. In this video, you will be seeing about these two different techniques. So first one is autoradiography, second is precursor product sequence. So here we are studying about tracer techniques, and four different types of tracer techniques are there. So one is autoradiography, second one is precursor product sequence, third one is competitive feeding, and the last one is sequential analysis. Clear? So first we can see about autoradiography. So it is one of the different types of tracer technique. Clear? So in autoradiography, detection of the radioactive isotopes in biological and other membranes were happening. Okay. So autoradiography is used in the detection of radioactive isotopes in biological and other materials. Clear? So this radioactive isotopes, first you have to understand the thing that these compounds we are directly feeding. So in the plant as such, we are feeding this one. So this autoradiography is used in the detection of radioactive isotopes in biological as well as other materials. Clear? And this technique is used for the study of whole morphology as well as histology of leaf or any other sample for their metabolic activity. So in the last semester pharmacognosy, we studied that plants were having lot of chemical constituents, and these chemical constituents were not entirely distributed throughout the plant, and it is sometimes seen in only in some part of the plant. For example, some compounds were seen in leaf, some in root, bark, etc. Okay, so here with the help of autoradiography, it is used in the study of whole morphology as well as histology of leaf or any sample for their metabolic activity. So here the metabolic activity, through this metabolic activity only lot of compounds were synthesized, and ഇതിനെയാണ് pathways എന്ന് പറയുന്നത്. ഇത് metabolic activity ആണ് pathways ഉണ്ടാവുന്നത്. So അതാണ് നമ്മൾ ഇവിടെ പഠിക്കുന്നത്. Clear? So autoradiography is used in the detection of radioactive isotopes in biological as well as other materials, and is used in the study of whole morphology as well as histology of leaf or any sample for their metabolic activity. Clear? Okay. So here a metabolite or a DNA of the sample. Okay. So ഒരു പ്ലാന്റ് നമ്മൾ എടുക്കുന്നു, അതിനകത്ത് ഒരു മെറ്റബോളൈറ്റിനെ സെലക്ട് ചെയ്യുന്നു, അല്ലെങ്കിൽ DNA. A plant means a metabolite or a DNA is radio labeled by a substance that emits either beta or gamma radiation. That is, so first we are selecting a metabolite of a plant or a DNA of a plant, and this thing is labeled by the help of some radioactive isotopes, and these compounds were having the capacity to emit either beta radiation or gamma radiation. Clear? Now, and after introducing this radio labeled compound, then an X-ray or photographic plate is directly placed on the labeled tissue. So ഒരു particular tissues നെ ആയിരിക്കും നമ്മളിവിടെ ലേബൽ ചെയ്യുന്ന ആ ടിഷ്യൂലോട്ട് ഒരു എക്സ്റേ അല്ലെങ്കിൽ ഒരു ഫോട്ടോഗ്രാഫിക് പ്ലേറ്റ് നമ്മൾ വെക്കുന്നു. Clear? Okay. Then, so it may emit radiation, and that is known as autoradiograph, and this recorded autoradiograph should be taken out and should be studied. From this, the biosynthetic pathway can be studied, or means get an idea about the biosynthetic pathway. So autoradiography, this technique is used in the detection of radioactive isotopes in biological as well as other materials. Clear? And autoradiography is normally used for the studying of whole morphology as well as histology of leaf or any other sample in the purpose for their metabolic activity. So why we are studying metabolic activity? Because through this metabolic activity only the secondary metabolites as well as primary metabolites are produced. And the secondary metabolites were having medicinal importance, and that is why we are studying about this metabolic activity. Clear? So here a metabolite or a DNA is radio labeled by a substance that can emit either beta or gamma radiation. Clear? That is a radioactive isotopes. So a metabolite or a DNA is radio labeled by a substance that can emit either beta or gamma radiation, and then an X-ray or photographic plate is directly placed on the labeled tissue. Now, autoradiograph is obtained. Autoradiograph is an image only, and this image is having the recording of this radiations. The radiations വന്നിട്ട് അവിടെ ഉണ്ടാവുന്ന റെക്കോർഡിനെയാണ് autoradiograph എന്ന് പറയുന്നത്, and this autoradiograph is recorded and it is studied, and from this you will be getting an idea regarding the biosynthetic pathway. Clear? So ഇനി ഇത് എങ്ങനെയാണ് ചെയ്യുന്നത്, അതാണ് മീൻസ് അത് എങ്ങനെ എടുക്കുന്നു എന്നുള്ള കാര്യമാണ് നമ്മൾ ഇപ്പോൾ പറയാൻ പോകുന്നത്. So here the tissue of the labeled plant is rapidly frozen. So after insertion of this photographic plate, and you have to keep it for some time, then you have to take the tissue of the labeled plant, and then it should be rapidly frozen, then it should be freeze dried, then it should be embedded in paraffin and sliced into thin sections with the help of a microtome. Clear? So first a radio labeled compound is introduced into the metabolic part of a plant. Then then a photograph means graphic plate should be inserted there. Then this this radio labeled compound will be having the capacity to emit beta or gamma radiation, and this may attach to the photographic plates, and an image is obtained, and this is known as the autoradiograph. And after this, autoradiograph the tissue of the plant is labeled plant should be rapidly frozen, then it should be freeze dried, and after that it should be embedded in paraffin and sliced into thin sections. Okay, now this section should be coated with a film of photographic emulsion. So this section should be coated with photographic emulsion. Then when it is exposed to radiation, when the tissues are exposed to radiation, the rays got recorded in a film, and when the films is developed, then silver grains will appear on those areas where the labeled molecules are present in the tissue. Clear? So ഇങ്ങനെയാണ് ഇത് ഡെവലപ്പ് ചെയ്യുന്നത്. ഒരു സാധാരണ ഒരു ഫോട്ടോ ഡെവലപ്പ് ചെയ്യുന്ന അതേ ടെക്നിക്ക് തന്നെയാണ് ഇതിനകത്ത് പറയുന്നത്. So ഇവിടെ ആ section should be coated with photographic emulsion, then it should be exposed to radiation, then this rays may recorded in the films, and when the film is developed, you will be getting some silver grains on the this photographic film or the X-ray film, and where means silver grains എവിടെയാ കിട്ടുന്നത്, labeled molecule ഉള്ള ഭാഗത്ത് silver grains കിട്ടും. Okay, and this technique is widely used for the study of sugar translocation and also for the determination of tissue or cellular location of the isotopes. അതിനു വേണ്ടിയാണ് ഈ ടെക്നിക്ക് യൂസ് ചെയ്യുന്നത്. ഏത് ടിഷ്യൂവിൽ പ്ലാന്റിൽ നമ്മളത് ഫീഡ് ചെയ്യണം, ഈ ഐസോടോപ്സ് മീൻസ് ആ പ്ലാന്റ് ഫീഡ് ചെയ്യണമെങ്കിൽ തന്നെ ഏതെങ്കിലും ഒരു പോർഷനിൽ മാത്രമല്ലേ നമുക്ക് ഉദ്ദേശിക്കുന്ന കോമ്പൗണ്ട് ഉണ്ടാവുള്ളൂ. അപ്പൊ അതുകൊണ്ട് തന്നെ ഈ സാധനം നമ്മൾ ഫീഡ് ചെയ്ത സാധനം ഏത് സെല്ലിലാണോ ടിഷ്യൂലാണോ ഉള്ളത് എന്നുള്ള കാര്യം അറിയാൻ വേണ്ടിയും ഷുഗറിന്റെ ട്രാൻസ്ലൊക്കേഷന് വേണ്ടിയുമാണ് ഈ സാധനം നമ്മൾ ചെയ്യുന്നത്. ഇതാണ് ഓട്ടോറേഡിയോഗ്രഫി എന്ന് പറയുന്നത്. ഓക്കെ, സോ ഇറ്റ് ഇസ് എ സിംപ്ലസ്റ്റ് ഓഫ് ഓൾ ദ ട്രേസർ ടെക്നിക്സ്. ഇൻ ഓട്ടോറേഡിയോഗ്രഫി എന്താണ് നടക്കുന്നത്? It is used for studying the entire morphology or histology of leaf or any other part of plant. So here a metabolite or a DNA is labeled, and then an X-ray or photographic plate is introduced there. Then this labeled compound is having the capacity to emit radiations, and it is recorded, and that is known as the autoradiograph. Now here the tissue of the labeled plant is rapidly frozen, then it should be freeze dried, then that should be embedded in paraffin and sliced it into thin sections. Okay, now these sections are to be coated with a film of photographic emulsion, then it should be exposed to radiation, then the rays got recorded in the film, and when this film is developed, silver grains can be obtained or seen on the photographic plate where the labeled compounds are located or labeled molecules are located. From this you can identify that where this one is present, that is in which cell or tissue this labeled compound is present. So this technique is used for the study of sugar translocation and also in the determination of tissue or cellular location of the isotopes. Clear? Now, we can see about the next tracer technique, that is precursor product sequence. This is a simple technique. Precursor product sequence. പഠിക്കാൻ simple ആണ്, but ചെയ്യാനായിട്ട് practically വളരെ difficult ആണ് precursor product sequence എന്ന് പറയുന്നത്. So precursor product sequence എന്ന് പറഞ്ഞാൽ വേറൊന്നുമല്ല. So for the formation of a compound, lot of means a starting material is there as well as lot of intermediates were also there. Then only you will be getting the final product. And the compound which is just preceding to the final product is known as a precursor. Clear? So here in this technique, a precursor of a constituent is presumed and its labeled form is introduced into the plant. So എന്ന് പറഞ്ഞാൽ ഇവിടെ ഒരു കോമ്പൗണ്ട് നമുക്ക് കിട്ടുമെന്ന് അറിയാന്നുണ്ടെങ്കിൽ ഉണ്ടാവുന്ന, ഇപ്പോൾ എക്സാമ്പിൾ ഒരു ആൽക്കലോയിഡ് ആണെന്നുണ്ടെങ്കിൽ അതിൻ്റെ ഒരു പ്രീകേഴ്സറിനെ നമ്മൾ അസ്യൂം ചെയ്യണം. എന്നിട്ട് അതിനെ ലേബൽ ചെയ്യുന്നു, അതിൻ്റെ കൂടെ ഒരു റേഡിയോ ആക്റ്റീവ് ഐസോട്ടോപ്പും കൂടെ കൊടുക്കുന്നു, എന്നിട്ട് അത് പ്ലാന്റിലോട്ട് ഇൻട്രൊഡ്യൂസ് ചെയ്യുന്നു. Okay, so here a precursor of a constituent is presumed and its labeled form is introduced into the plant. Then after some time the constituent is isolated and purified and its radioactivity is measured. Clear? So in precursor product sequence, a precursor of a constituent is presumed and its labeled form എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ റേഡിയോ ആക്റ്റീവ് ഐസോട്ടോപ്പും കൂടെ ഉള്ള ഫോമിനെയാണ് ഇവിടെ ഉദ്ദേശിക്കുന്നത്. അത്രേ ഉള്ളൂ കേട്ടോ. So a precursor of a constituent is presumed and its labeled form is introduced in the plant, and after some time the constituent is isolated and purified and its radioactivity is measured. Clear? Okay. And this technique won't give much information because the compound may enter the general biosynthetic pathway and it may get distributed randomly among the various phytoconstituents. So here എന്താണ് സംഭവം എന്ന് പറഞ്ഞാൽ, here we are just feeding only a precursor. So you knowing that a final product, ഈ final product ഉണ്ടാവുന്നതിന് മുമ്പുള്ള ഒരു പ്രീകേഴ്സറിനെ മാത്രമാണ് നമ്മൾ ഇവിടെ ഫീഡ് ചെയ്യുന്നത്. സോ ഈ ഫീഡ് ചെയ്യുന്നതെന്ന് പറഞ്ഞാൽ ലേബൽ ചെയ്തിട്ട് അതിനകത്ത് പ്ലാന്റിനകത്തോട്ട് കയറ്റി വിടുന്നത്. അപ്പോൾ ഇത് ആ ഒരു സീക്വൻസിൽ തന്നെ പോകണമെന്നില്ല. ഈ മെറ്റബോളിക് പാത്വേയിലുള്ള എല്ലാ കോമ്പൗണ്ടുമായിട്ട് കയറി ഇന്റർഫിയർ ചെയ്യും. അത് കാരണം തന്നെ കൂടുതൽ ഇൻഫർമേഷൻസ് നമ്മൾ ഈ ഒരു ടെക്നിക്കിൽ നിന്ന് എടുക്കാൻ പറ്റൂല. ക്ലിയർ? So this technique won't give much information because the compound may enter the general biosynthetic pathway and gets distributed randomly among the various phytoconstituents. Clear? So here in precursor product sequence, a precursor of a constituent is presumed and its labeled form is introduced in the plant, and after some time the constituent is isolated and purified and its radioactivity is measured. Clear? And this technique won't give much information because the compound may enter into the general biosynthetic pathway. Specific ആയിരിക്കില്ല, ആ പ്രീകേഴ്സറിൽ മാത്രമല്ല അത് പോയി പിടിക്കുന്നത്. It may enters into the general biosynthetic pathway and gets distributed randomly among the various phytoconstituents. Okay, so in such cases what we can do? Further proof can be obtained by double or triple labeling. സോ ആദ്യം ഒരെണ്ണം ലേബൽ ചെയ്യുന്നു, അപ്പോൾ അതല്ലാന്ന് മനസ്സിലായി കഴിഞ്ഞാൽ രണ്ടാമത് ഒരെണ്ണം ലേബൽ ചെയ്യുന്നു, അതും അല്ലാന്ന് മനസ്സിലായി കഴിഞ്ഞാൽ മൂന്നാമത് ചെയ്യുന്നു. ഇങ്ങനെ ചെയ്തു കഴിഞ്ഞാൽ ഡബിൾ ലേബലിംഗ്, ട്രിപ്പിൾ ലേബലിംഗ് ഇതൊക്കെ ചെയ്തു കഴിഞ്ഞാൽ നമുക്ക് ഈ പ്രീകേഴ്സർ ഏതാണെന്ന് എക്സാക്റ്റ് ആയിട്ട് നമുക്ക് കണ്ടുപിടിക്കാം. സോ ഇവിടെ നമ്മൾ പറഞ്ഞ ഒരു പ്രീകേഴ്സറിനെ നമ്മൾ പ്രസ്യൂം ചെയ്യണം അല്ലെങ്കിൽ അസ്യൂം ചെയ്യാണ് ചെയ്യുന്നത്. അസ്യൂം ചെയ്ത പ്രീകേഴ്സറിനാണ് ഇതിനകത്ത് കൊടുക്കുന്നത്. അതിന് മുമ്പുള്ളതാവാം, അല്ലാതിരിക്കാം. സോ ആണെങ്കിൽ തന്നെ ഇത് ജനറൽ ആയിട്ട് ഈ പാത്വേയിലോട്ട് അങ്ങ് പോവുകയാണ് ചെയ്യുന്നത്. അതുകൊണ്ട് മറ്റ് ഇൻഫർമേഷൻസ് കിട്ടൂല. അങ്ങനെ വരുമ്പോഴേക്കും ഡബിൾ ലേബലിംഗ്, ട്രിപ്പിൾ ലേബലിംഗ് ഒക്കെ ചെയ്തു കഴിഞ്ഞാൽ ഈ ഒരു ടെക്നിക്ക് വഴി ആ പാത്വേ കറക്റ്റ് ആയിട്ട് നമുക്ക് എടുക്കാൻ പറ്റും. ക്ലിയർ? So that is about precursor product sequence. Okay, so in tracer techniques, അപ്പോൾ ഈ വീഡിയോയിൽ എന്തൊക്കെയാണ് ഉണ്ടായിരുന്നത്? What are the different methods involved in the elucidation of biosynthetic pathways? So അത് tracer techniques ഉണ്ടെന്ന് പറഞ്ഞു. പിന്നെ അതേപോലെ വേറെ എന്തുണ്ടായിരുന്നു? ഗ്രാഫ്റ്റിംഗ് ഉണ്ട്, then isolate mutant strains ഉണ്ട്. So ഇതൊക്കെ അതിനകത്തുണ്ട്. അതിൽ tracer techniques എന്താണെന്നുള്ള കാര്യം നമ്മൾ പറഞ്ഞു. It is a most simple technique. So here radio labeled compounds are introduced into the plant. So why we are selecting this radio labeled compound? Because they were having the capacity to retain its radioactivity even after എന്താണ്? So even after incorporating into the chemical reactions. So അതാണ് പറഞ്ഞത്. ഇനി അതിനകത്ത് രണ്ട് ടെക്നിക്സ് ആണ് നമ്മൾ ഇതിനകത്ത് പഠിച്ച. ഫസ്റ്റ് is autoradiography and second thing is precursor product sequence. അത് നിങ്ങൾ ഒന്നും കൂടെ ഒന്ന് നോക്കിക്കഴിഞ്ഞാൽ നിങ്ങൾക്ക് ഇത് ക്ലിയർ ആയിട്ട് കിട്ടുന്നതാണ്. ഓക്കെ, അപ്പോൾ ഇത്രയാണ് ഇതിനകത്തുള്ളത്. സോ ഇതിന്റെ ബാക്കി ടെക്നിക്സ് എന്ന് പറഞ്ഞാൽ അടുത്ത വീഡിയോയിൽ നമുക്ക് കാണാം. ഓക്കെ, then thank you.